So basically, as I mentioned, the replication of findings is a current and ongoing emphasis at NIH, and this is what I just said. And so, as Elena pointed out, but we're going to talk about it in a slightly different way now, it's the requirement of every investigator then to safeguard against misconduct and to build a laboratory in which scientific integrity and reproducibility of data are the central focus. So basically, there are several factors that we can think about that lead to findings that are not replicable. And these factors are closely related to, I think, our day-to-day approach to scientific integrity. So I wanted to start, very often RCR sessions are all about these egregious cases. Elena had one slide about them, and I'm going to also have one slide about them. These are a couple of cases actually from our very own field. Luckily, these are extremely rare, but I think start with them as kind of the quintessential example of difficulty with replicability when misconduct occurs, falsification of data, fabrication of data, plagiarism. We all know about these. We do not conduct these deliberately. There are a couple of major cases in our discipline. One of them, many years ago, a grant on orofacial motor control impairment in Parkinson's, where, and this is a quote from the NIH response to this, where this researcher was engaged in scientific misconduct by falsifying and fabricating certain figures and research results supported by NIH grants. So that's a really extreme example, making up data. A second example has had a lot of prominence recently, Mark Hauser's case. And this is a quote from Hauser. Although I have fundamental differences with some of the findings, Hauser wrote, I acknowledge that I made mistakes. I let important details get away from my control. And as head of the lab, I take responsibility for all errors made within the lab, whether or not I was directly involved. So this is, again, this idea of the role of the PI being ultimately responsible. And in both of these cases, I think it's worth pointing out that the whistleblowers, and this is as it goes, tend to be students or postdocs. So talk about a high level of vulnerability. These are the last people who you want to be stuck in this situation of blowing a whistle on their mentor. It's a pretty nightmarish situation. So that is the extreme. We are opting to not talk about the extreme today, but I think it's kind of a good starting point. So we want to think about, in our labs, what do we need to guard against, kind of grading down from this extreme. How can we think about replicability in a responsible environment? One thing that we have to think about, in my opinion, we'll open it up to you in a few minutes to get your ideas here, but extreme commitment to a particular theory or result can really have a negative impact on replicability, for example. And I'm kind of focusing on replicability or conduct as a little bit overly interchangeable, perhaps. This may lead to bias in how data are obtained and or are interpreted. And all of us have, a passionate adherence to a theory, I would guess. But again, it's something that we need to really guard against as we collect our data and analyze our data and think about our data and train our students to think about our data. Bias is rampant. We know this. This is why NIH is actually extremely engaged in this process right now. And something else that might feed into this problem is the well-known fact that it is very difficult to publish negative results. We have ginormous rewards to really exciting big findings and very few rewards for negative results. So in a way, things are very stacked against us to at least subtly engage in a very subtle form of potentially misconduct. I want to be careful with my terms here, but you see the point, right? So that's kind of the theoretical, how your theoretical biases and frame can influence your work. Experimental issues can also lead to problems of replicability, which is the crucial, the core of what we do. So poor experimental design, lack of blinding or lack of randomization, lack of appropriate controls in our experiments, both in terms of participants, in terms of tasks, cherry picking of participants and results. So when we exclude participants, when we select the figures to put into a paper, I think these are familiar issues for all of us. How are we thinking about that in terms of, again, scientific conduct? Incomplete descriptions of analyses and statistics and sample sizes and lack of inclusion of details. And obviously, our journals all force us to page limitations. Our journals, our reviewers want us to write very readable, accessible papers, and yet there's a price to pay, perhaps, in terms of other people replicating our work down the road. So what are granting agencies and journals doing about this? And I got some of this, I should say, speaking of potential plagiarism and not citing from the a presentation that was shared with us by Dan Sclair from the Deputy Director of NIH, who was really talking about, so what is NIH trying to do about this? So one thing that I think might be coming down the road at NIH based on at least what people are talking about, is increased systematicity in the review process regarding how some of these replication issues might be handled. Rewarding the idea of attempting to reward replication studies and negative results, pressure to do this, and pressure to encourage journal editors to allow researchers and require researchers to include more detailed method sections. So these are at least some things that people are talking about that could be coming along to try to address some of these issues. So basically, with that kind of framework about replicability being kind of the day-to-day goal that we have, obviously I think replicability begins in each of our labs. And creating an environment that is conducive to maintaining the highest standards of scientific integrity is, of course, the job of the PI again, as is protecting our participants. I thought, I don't know if this will quite work, I'll give you the idea, but I thought what we would do is take just a minute or two for each of these, and again, a kind of more active way to think about this is for your lab, the lab you work in or your own lab. The one time that I think, or there are a few windows or a few places we really need to think about how do we build this environment, and one of those is when a new person is entering our lab. What are some things we can do? And then I have some ideas from my lab, but let's first open it up to you. We're good enough on time. I think give you a minute or two at your tables once again, and I won't go around to every table because I'll randomly pick a couple tables for each one of these. So be prepared. What is something you can do in your lab to set up an environment so data, right now we're talking about the responsible formulation of theory and applying that theory to your lab and how you collect and analyze your data. That's kind of the issues that we're primarily thinking about. So I'll give you a second. Does that make sense? So for your own labs, what's a good way to set that up?
Yeah. Okay. So I do EEG, so there are a lot of judgment calls with deciding which trials you're going to throw out and which trials you're going to keep, because if the person has done that, then you have messy data. But sometimes you have like small eye movements that could actually be an ERP component. So we talked about how having being clear about how many trials you're keeping, having standards that you set before the experiment or before the data is analyzed, and then also having it, the artifact detection may be done in parallel by two different people in the lab and comparing.
Thank you. Thanks. Is there one more who'd like to? I'll let you choose. One more? You guys sounded like you were really talking on this.
So one of the things we do in my lab is that we have an internal logbook that is computerized, that is all on the same server, so I expect all the students to continuously put in their logging time. Another thing that sort of keeps people on task but also ensures that people are doing what they're supposed to do with regards to data entry is that I have my students write a weekly progress report and talk about the data that they collected. Yep.
They must love it every week, but... Okay, great. So hopefully I'll get everybody by the end here. So some ideas from my lab. we have orientation for all new members. RCR training goes without saying, but we really have very explicit instruction in data analysis and documentation. We do have manuals. You have your manuals online. We have really systematic manuals that anybody walking into the lab who is just starting, they do read them. And we revisit them at our lab meetings regularly so that we co-write them as a group on about 6 to 12 month intervals to update and make sure we are not drifting. And these are also the way when people walk into the lab, they're first learning about how we cue kids, how we analyze data, all of these factors. So we actually really rely on the manual. We also, every new person, we build little teams that are responsible for each data analysis. And an experienced research assistant, a doctoral student, a postdoc, a research associate is in charge of each team, is in charge of training in that team, and deals with micro-level reliability issues with me, and then brings them back to our a weekly lab meeting is, we feel, crucial. And those are just some of the examples from my lab, I think, yeah. So it's very shared accountability for every acquisition and analysis. So any other kind of general, these are just kind of from what we try to do. So any brand new person has a reliability bar they have to cross. Very good. They don't code data. That's a great idea. So good. Great. So the next issue is ongoing, and ongoing management may be similar enough. I think what I may do is talk about my examples and then open it up just once more in the interest of time. So ongoing data acquisition and analysis is it's again really regular lab meetings where we take turns. We have many sub-projects, and we take turns that the team involved in that sub-project is presenting it and bringing in data analysis or queuing. We do a lot with, an issue we hit a lot, for example, is we test language impaired children, and we are getting them to produce different kinds of sentences and words and things like that. And so we have very rigid rules about how you can cue them, how many times you can cue them, so that our language impaired kids get the same cues as our typical kids. So what is happening with the queuing over a 2 or 3 year study? What is happening with the data analysis? So we just have these rolling reportings where people bring their work to our lab meeting. Also, when there are any difficult data from a given child, those always are every single lab meeting. Any data problems this week? And we have them bring pictures, and that everybody looks at together and comes to consensus for difficult to analyze problems. And again, our lab manuals are where we write all of these down so that they're documented. People do not read them regularly, but they do annually. And I'd love to hear when we break out again, I still keep written lab notebooks. You have electronic. And the reason I do that is writing things down while you're doing it in pen is really different. It could just be my age from handling things electronically. So I'd love to get away from the paper, but when it comes to real data analysis issues, I find being able to go back to the notebooks, see what people did, and be able to replicate that. We do that a lot, and I am still wed to paper for a little portion for that reason. Paper and pen, not pencil. Obviously, maintaining records of all research participants where we collected data or where they decided not to participate or where we decided not to include them, to be able to address that cherry-picking problem I alluded to, and maintaining well-labeled files with data. So from our data notebooks to our videos to our actual data files for a very extended time period. So all of these relating to replicability. Documenting processes in our research, the steps that we use to arrive at outcomes. This is in a way what our manuals, what our data notebooks are about for any new trainees, for new people as they roll in, clarifying procedures, standing up to an audit. That is really what we're thinking about. But the main thing we're thinking about for our everyday science is our studies take years at times, usually, and we want who we run in year 4 to be the same as who we ran in year 1. That's absolutely crucial, and that is really a huge rationale, but an added benefit is if anybody looked or anybody wanted to replicate, as if they ever would, that we could really stand up to that. So that is, and then the third issue is theoretical framing of findings. So we've talked a little bit about new lab, about approaches to handling data in the lab. What about the situation where, which happens to me a lot, which is why maybe I'm highlighting this one, where my data don't fit my hypothesis, where the story is different? How do you handle that? And so give you a second maybe to either add to the prior or to think about this third issue of, if your data don't fit your theoretical account, how do you handle that in your lab?
I have a tough one. So we were talking about how if you have a science question, if you're very clear with students about there being multiple correct answers and how everything, you just let the data lead you, then that is a reasonable way to go forward. But if you're not asking a science question, if you're developing a new tool or developing a new algorithm, then it's very clear what a performance spec would be, which means that you can't pretend like doing worse than something is still great. So it's difficult to think about the theoretical framing of poor findings in that case, and it seems like a long-term problem.
There you go.
Well, I guess we talked a little bit about making sure that it's really a change in your theoretical concept versus an outlier in your data or a problem with your data. So going back and checking the original raw data to make sure there wasn't, a miscalculation or something that caused it to be outside of what you hypothesized it to be.
Go for it.
Oh, Julius was saying that one of the things he does is that he goes into everything basically prepared not to publish the results, because if they, and or revising your theory about what's going on.
And we also just talked about, that's happened. We also talked about not just whether there's an outlier that's causing the problem, but whether your measure is not sensitive enough, and that's important. So that
outliers are informative. Noise in data is informative, and this drive to have non-noisy data non-noisy human data is just irrational. Then it's really, how you tell a story about the data as they are without throwing, one outlier, sure. But, if you've got multiple outliers, then there's a different story there, and to be sensitive to that.
Just a couple of quick things that we do. I think these were really deeper examples than mine. Again, our teams, I think basically we talk a great deal about that the data need to tell the story. I have so many anecdotes about some of, frankly, my own favorite work from my own lab that was completely counterintuitive and did not work with my hypothesis, that I always make sure that I introduce early and often as, because doctoral students, when they're first, their study comes out and we run the stats and it's like, oh no, I'm like, this is not over. This is the beginning of the interesting part. We are seeking the truth. We are trying to, if we knew already, it wouldn't have been worth doing this experiment. So basically, sort of trying to reframe a little bit about how the theory and the data interact is something we do. So just to finish up and let Mario have his turn, above all else, your job, what you have to do is model the behavior you expect from students in your lab. You need to stay close to the data and to the processes in your lab, and to go to our very sad Mark Hauser story. You're responsible, if it's because you were absent or if it's because you did the wrong thing.